The first skeletons you will probably recognize in the image to my left are the ones you would find in the human body. The skeleton you are less likely to recognize, but is also found in the human body, is the cytoskeleton. You are actually looking at the skeletons of neurons, or more specifically, the microtubules of neurons. Microtubules are members of the cytoskeleton system, and similar to the function of bones in our body, they are responsible for giving cells their size, shape, and structure. They also act as railways to transport organelles like mitochondria, which supply energy to the cell, and vesicles, containing neurotransmitters and receptors which ultimately contribute to neuronal cell-to-cell signaling via a neuron's axon. Axons are what get damaged after a nerve injury, thus preventing sensation and motor control. In this immunofluorescence image, you can see the microtubules in adult dorsal root ganglion neurons. These neurons are responsible for touch, pain, and proprioception. They are the only adult neurons capable of being cultured, and they are unique peripheral neurons that only extend axons. The hair-like processes in the image behind are axons made visible by immunolabeling neuronal microtubules with fluorescent dyes. I use these neurons to study microtubule-mediated neuroregeneration. I, along with a growing number of researchers, believe that unique microtubule-associated proteins in small molecules can promote axons to regenerate. It is here that the metaphor between microtubules and our skeleton works again. Just like our bones, people have a misconception about microtubules and think of them as rigid, unchanging, and stone-like. This couldn't be further from the truth. Microtubules are highly dynamic and inherently polarized. A single microtubule has two general domains: a stable domain that is older and resists depolymerizing, and a labile domain, where tubulin is added and constantly undergoes bouts of growth and retraction, just like the bones in our body. These microtubule dynamics are paramount for axon growth. Recently, microtubule-associated proteins in small molecules have been found to affect these labile domains of microtubules, thus regulating their dynamics and increasing microtubule mass to promote the regeneration of axons. The addition of microtubule mass to the regenerating end of an axon is essential. Microtubules must be engaged to elongate an axon. A growing axon forms a structure, called the growth cone, which is responsible for producing new axon. Microtubules are required to engorge the growth cone to move it forward. Microtubules again can be compared to the bones of our body. To heal a broken arm, new bone must be created. You can't have axon regeneration without adding more microtubule mass. This is an exciting, novel, and hypothesis-driven approach to nerve regeneration that can hopefully aid people suffering from nerve injuries who have no available treatment to promote regeneration.